Hello, this will be talk number 9 in the series from Principles of Biochemistry, written by Albert Lehninger, 7th edition. Today we're going to discuss protein structure and discuss how the amino acids link together and what they form. So when we talk about protein structure, we're looking at 4 different levels of protein structure. The first of these is what's called primary structure. This is a sequence of amino acids. This is the sequence of amino acids, the 20 that we've discussed before, once you link them by elimination of water, giving the peptide bond. So you can have a primary structure of, say, alanine, then valine, then glutamine, then tryptophan, then lysine, and proline in that order. The secondary structure deals with the conformation of the amino acids within the sequence. Again, these proteins, as they're formed, will fold on each other, and they fold according to the characteristics primarily of the structure of the protein, the amino acid structure, but also the surrounding physiologic conditions, namely temperature, salt concentration, and the impact of various constituent groups that may be a post modification. So we talk about different types of secondary structures. There's the key one is alpha helix, and then there's also beta sheet, and we'll be showing those shortly. Thirdly, there is a tertiary structure, and this is a three-dimensional conformation conferred by folding of the chain upon itself. So as the strand of the amino acid sequence occurs, it will fold on itself, and this is why you get proteins that, if you look at them under high resolution, they have sort of a globoid, globular appearance, and they're not particularly round, but they have different zones and different domains, and the domains convey a certain form of a certain function to them, depending on whether they are regulatory or if they're involved in catalytic action as an enzyme. The quaternary structure, then the interactions of those particular globoid conformations. You have protein dimers, trimers, and tetramers, such as hemoglobin. Again, the protein function follows form. Mutations affect the amino acid sequence, its structure, and function. The most famous of this, of course, being sickle cell anemia, which is we'll be covering that shortly. That's a single point mutation with a change of amino acid. Today we derive protein sequence from the DNA genome. We have an encoding sequence, but in the previous time we would serially remove the amino terminus in a protein, and with an automated process you can involve maybe small molecules up to 40 amino acids in length. So you literally remove the amino terminus from this small chain and determine what the sequence was. If you had larger proteins, you'd have to break the disulfide bonds, and those would be the molecule cysteine, which is cysteine involving elimination, and a ester formed with the terminal sulfide linkages. Then you would break those peptides with proteases, and these proteases are specific to certain sites, and we'll be showing those shortly. So today we can use the DNA genomic database and then get the protein sequence through that. Once we know that structure of the genome, we can find out through the codons what the from the RNA transcript what the sequence is. But in the olden days, which weren't that long ago, you had to literally remove the amino terminus. You'd have to take the protein, break it up, and use a protease to find out. So some of these are found in the body, the human body, and they're used quite extensively to break down foodstuffs. But we talk about trypsin. We talk about trypsin breaking a protein so that lysine and arginine are at the carbonyl site. We talk about chymotrypsin. These are both pancreatic enzymes. They will break so that the phenylalanine, tryptophan, and tyrosine are at the carbonyl site. There's also Staph aureus protease. Staph aureus would give a very very extensive destruction of tissue if you have staph in the lung or a soft tissue wound. There's a protease there, and it breaks aspartate and glutamate at the carbonyl site, so it will act so that the residual protein will have either aspartate or glutamate at the carbonyl end. There's also pepsin, which is also part of the gastric fluid in the human, and that would break so that you have leucine, phenylalanine, tryptophan, or tyrosine at the amino site. We can take mass spectroscopy as a sequencing tool, but it's limited because the macromolecule dissolution in the gaseous phase is something that is an inherent limitation. So when we have the solution to that particular limitation, mass spectroscopy is to ionize the macromolecules with laser light and then subject micro droplets to electrical charges and disperse the solution, evaporate. So you've got ionically charged macromolecules in a gaseous phase. That is one way to get around the limitation of doing protein sequence with mass spectroscopy. You can first treat the macromolecule with protease, and then you get two mass spectroscopy analyses performed in series, before and after. The first series splits of short peptide sequences, and the fragments are further fragmented by high energy collision with helium or argon. And then the second one would break down peptides further, and then you get the sequence elucidation from that. And if you had small samples of protein, you can utilize this quite nicely. So this is an example of that particular technology, and where you get electrospray ionization, the separation with collision cells, and then the second phase here, so you get separation and breakage, which are all detected, and then you get, for example, an amino acid sequence here with a breakage of this peptide right at this point with these ionized fragments, and those would go into the unit, and you would get the fingerprint right here, which would be characteristic of this amino acid sequence here. Again, this is in bioinformatics coding. For protein synthesis, there are three ways of doing it. There's purification from tissue, there's chemical synthesis, and there's genetic engineering. So the chemical synthesis requires anchoring of one end to a solid support resin. The amino acids are added one at a time. You can form roughly 100 amino acid peptides in a day. The nucleic acid synthesis is the same methodology. You have an anchoring point, and then you're adding one nucleic acid at a time to that. The protein domain is an area of critical function which is greatly impacted by mutations in inhibition or activation. I discussed, for example, the example of hemoglobin, which has a specific domain where indices which can impact that site or critical imports, and we'll be talking about hemoglobin at some future point. The protein sequence is conserved between closely related species. They diverge with time, and you can also have horizontal gene transfer, which where the entire sequence will transfer between two organisms. And this is an example, for example, of antibiotic resistance. So when you're looking at the eukaryotes and archaea and bacteria, the mitochondria can have movement across kingdoms to the eukaryotic line. Chloroplasts can be incorporated in certain areas like euglena and trypanosomes, and then higher forms. These higher forms will have mitochondria as organelles located within the cell, so they are essentially symbiotic remnants of bacterial species, with their own particular sequence characteristics. That will do it.